Chapter 1. DNA Fingerprinting. Also known as DNA profiling, is a technique used to identify individuals based on their unique DNA characteristics. It has applications in forensic science, paternity testing, genealogy, and even wildlife conservation. Here's a breakdown of the key points about DNA fingerprinting. Chapter 2. Basics of DNA structure. DNA, or deoxyribonucleic acid, is the molecule that carries genetic information in all living organisms. It's made up of a sequence of nucleotides, each containing one of 4 nitrogen bases: adenine, cytosine, guanine, and thymine. DNA is almost identical across all humans, but there are small differences, particularly in regions that don't code for specific proteins. These differences make each person's DNA unique, which is what DNA fingerprinting focuses on. Chapter 3. How DNA fingerprinting works. DNA fingerprinting examines specific regions in the DNA sequence called short tandem repeats or variable number tandem repeats. These are regions where a short sequence of DNA is repeated multiple times, and the number of repeats varies from person to person. For example, in one region, one person might have A get repeated 5 times, while another might have it repeated 7 times. By comparing the number of repeats in multiple STR regions, a unique DNA profile can be created for each individual. Chapter 4. Process of DNA fingerprinting. 1. DNA collection. Samples can be taken from blood, saliva, hair, or any body cell. 2. DNA extraction. DNA is isolated from the sample using chemical processes. 3. PCR, or polymerase chain reaction. If the sample size is small, PCR amplifies the specific DNA regions so there's enough to analyze. 4. Gel electrophoresis. DNA fragments are placed in a gel and an electric current is applied. Since DNA is negatively charged, fragments move toward the positive end of the gel, with smaller fragments moving faster than larger ones. This creates a unique banding pattern based on fragment size, representing the STRs or VNTRs. 5. Analyzing the pattern. The banding pattern, like a barcode, is unique to each individual. By comparing the patterns between samples, analysts can determine if they match or differ. Chapter 5. Applications of DNA fingerprinting. 1. Forensic science. In crime investigations, DNA from crime scenes like hair or blood can be compared with suspects' DNA. If the patterns match, the DNA likely came from the same person. Paternity and ancestry testing. DNA profiling can confirm biological relationships, as children inherit half of their DNA from each parent. Patterns in the child's DNA should match the mother's and father's in predictable ways. 2. Conservation and wildlife management. DNA fingerprinting helps track genetic diversity in wildlife populations, identify species, and prevent poaching by identifying the origin of animal products. Chapter 6. Accuracy and limitations. DNA fingerprinting is highly accurate, especially when multiple STR markers are used. Modern techniques analyze around 13 to 20 STR regions, creating profiles that are statistically unlikely to be shared by more than one person. However, DNA fingerprinting isn't foolproof. Cross-contamination, human error, or degraded DNA samples can affect results. Also, while it can exclude a person as a source of DNA, it cannot guarantee that the DNA definitely came from a specific individual unless there's a perfect match. Chapter 7. Ethical considerations. DNA fingerprinting raises privacy concerns, particularly with storing DNA profiles in databases. There are also questions about consent, especially in cases where DNA is collected without a person's knowledge, such as from discarded items. Chapter 7. Summary. DNA fingerprinting is a powerful tool that relies on analyzing unique patterns in DNA to identify individuals. By focusing on STRs and VNTRs, it creates a fingerprint of a person's genetic material, making it incredibly useful in forensic science, paternity testing, and beyond. While very accurate, DNA fingerprinting does have limitations, particularly in cases where DNA quality is poor or handling isn't meticulous.